Well, today I'm going to present my experience developing a tetraploid map and also to find QTLs for the traits that we have been evaluating, which is calcium and tuber quality traits. So I've used a population that has been derived from Atlantic times superior. We chose these parents because Atlantic has high yield and very good chip quality, but it lacks of internal quality, and also it has low tuber calcium. And superior is a completely opposite for those traits, so it has low yield, low chip quality, but it has a very good internal quality and high calcium. So the presentation is divided in 4 sections. The first section is how I selected markers for mapping. The next one is how we go from having the markers to have a linkage map using Tetraploid map. And the next section is how to detect the QTLs after we have the map. And finally, I will mention the conclusions as a summary. Well, first, how to select your markers. We all, people that have been working with these SolCAP SNPs, we have received a data set with more than 8,000 SNPs. And the first time you open your file, you feel overwhelmed, as everyone else has mentioned before. And also you start feeling frustrated because you see that you don't move on really fast. It takes time to figure out how to select your markers. And especially your professor and your peers are always asking you, do you have a map already? And do you have a map? And you're just selecting markers still. So, but I see from the experience of the other graduate students that it's normal to have that situation. So I will show what I did in this case. I had 8,303 SNPs to begin with, and the population, I'm just putting the numbers of the progenies, not the parents. So the progeny was 184 genotypes. Then I removed 637 SNPs that were that had a quality label as questionable or bad. Then I also removed the SNPs that had 0 clusters, and those that had no hits, and those that were located in more than 2 positions in the genome, so more than 2 hits. And then I had a remaining set of 4,600, more than 4,600 SNPs in the same 184 genotypes. And I use this data set of to make a cluster analysis. And this cluster analysis is like a quality control for me, because this population has been grown for many years. It was developed in 2003, so it has been planted many years and manipulated. So I thought it may have some problems, and I actually found some. Well, the first thing you can see here is a principal components analysis. And in red, I have the Atlantic genotypes. And you can see that one of our parents was mislabeled, and it was labeled as superior, but it was actually Atlantic. Also, one of the progenies was actually Atlantic. And it's very clear here in the principal components analysis that they are the same genotype, but they have been labeled in different ways. And it's probably a consequence of having been grown for so many years. We have the other parent, superior, and there was one of the progenies that was actually superior instead of being a progeny. And all the real progenies are in between Atlantic and superior. So we can trust that those are part of the progenies and they can be used for the mapping analysis. But we have also 4 clones that are very far from what we would expect for a progeny. So I'm assuming that those belong to the other population that we have been growing together with this one, which is Snowden by Superior. Well, after I did this analysis, I could really just use those that are real progenies for my mapping analysis. And also all the clones that were Atlantics, I made just a summary and used just one. I took all those clones and just made it like one clone, one consensus clone for parent one, and also for Superior, those 2 clones that are Superior, I made a consensus and it was the parent 2. Another analysis I made is genetic distance analysis to find duplicated samples, and we had some samples that had been that we had sampled twice, so those clones were repeated, and we sent 2 samples to genotype, and I could see that in this analysis. And also we can see in the orange box that all those groups are clustering individuals that are very, very similar, so almost 0 distance. And we may think that those are just replications of the same genotype, even though some of them have different codes. But at this point, I'm not going to remove those genotypes because the data set at this point still has to be cleaned more. But that's what I could find at this point. The next steps I followed, I had 4,605 SNPs in 184 genotypes previously, from before the clustering analysis. And after the clustering analysis, I eliminated those mixed progenies that don't belong to this population, and also those that I knew were duplicated genotypes. So I eliminated 8 genotypes from the analysis. Then I removed all SNPs that had one or both parents missing, and those that were located in 2 positions in the genome. Then I just kept all the crosses that were nulliplex by simplex and simplex by nulliplex, and I removed all those SNPs that had more than 10% of double reduction. And after doing that, I had 962 SNPs remaining in 176 genotypes. And now I'm going to show a little bit of more detail on the double reduction analysis. So from the markers I had, I plotted how many individuals had a certain percentage of double reduction. So you can see that most, how many markers had the certain percentage of double reduction. So you can see that most of the markers had less than 5% of double reduction, but there were some that were above 10. And what we expect from the kind of cross that we have, nulliplex by simplex and simplex by nulliplex, we expect a 10% of double reduction, so as the highest. So everything that was above 10%, I just eliminated from the data set. So with the 962 SNPs and 176 genotypes, I put that data set into QTL. This QTL is a very useful software. It's developed for diploid populations, but it has very useful tools to clean up our data sets. So that's why I used it. And I eliminated using our QTL all duplicated markers. It means all markers or all SNPs that had exactly the same information. What the software does is it groups all those that have the same information and just keeps one as a representative of all others. Then I also eliminated the markers that had too high segregation distortion. It means that they don't fit well to the 1-to-1 segregation ratio expected. Then I eliminated markers that were typed only in very few individuals, and also the individuals that had very few SNPs genotype. And in the end, I got a data set of 619 SNPs and 166 genotypes. With that data set, I made a genetic similarity analysis. And what we expect from our progeny is that the genetic similarity should go from 0 to 1. And here I included the parents also. So the parents have a 0 genetic similarity because wherever parent 1 is nulliplex, the other one is simplex, and wherever the other one is simplex, the parent 2 is nulliplex. So they are completely different, so that's why it's a genetic similarity of 0. But we see also that we have some genotypes that had 1 of genetic similarity or very close to 1. Those should be eliminated to have a better analysis for our mapping purposes. So that's what I did in this case. I eliminated the duplicated genotypes with a more than 0.9 genetic similarity using RQTL. And my final data set was of 619 SNPs and 151 genotypes. And in this case, what the software, the RQTL, does is it groups the genotypes by similarity and keeps 1 as a representative of that. Well, this is a summary of my data set that I'm going to use for mapping. And you can see the position in base pairs and the markers and by chromosome. So this is based on the pseudo molecule. And this is what I would expect to have if the match between the position in the pseudo molecule is perfect to the position in the linkage map. So it's a ideogram here. This is in these graphs, you can see also the genotype frequency, nulliplex and simplex, and you can see it's very close to 50-50, which is what we expect. Well, next I'm going to show how we go from having our SNP data set with already selected markers for mapping to develop a natural linkage map. And this also takes time and a lot of try and error, especially learning how to use the Tetraploid map software. Okay, I use the Tetraploid map software. As you heard before, it has some limitations, but it works well. And the type of crosses I've used is simplex by nulliplex and nulliplex by simplex. The segregation ratio expected is 1-to-1. I also use the criteria to eliminate those markers that had too high segregation distortion with a p-value less than 0.001. And the markers that were in coupling with more than one homolog were also eliminated from the final map. Well, this is the summary of the procedure. First, we have to cluster the markers by chromosome. Oh, well, this is what I've done because I've seen that there are some differences with other people's mapping procedures, but this is what I think worked better for me and is very logical in my point of view. First, I loaded the whole data set, the 618 markers in Tetraploid map, and then I clustered by chromosome. Then within each cluster, I ordered the markers by parent. Then there was pairwise analysis of coupling and repulsion, and this is the step that takes the longest because you have to compare every 2 markers and see if they are in coupling or repulsion, and then assign them to a certain homolog. And finally, after you have assigned to all the 4 homologs, you can draw your map. And this map will be, you will have a map for each parent for each chromosome. So it needs 24, I think, 24 maps. Okay. This is a summary of the final map. I started with 424 markers for Atlantic and 195 for Superior, with a total of 619. And then I mapped 414 Atlantic. So you see there were 10 that were eliminated using those criteria that I just explained, high segregation distortion, or if they couldn't be assigned to just one homolog. For Superior, 187, and making a total of 601 markers mapped. And the length of the chromosome for Atlantic was 1,238, which is close to what we expect. We expect around 1,200, 100 centimorgan per chromosome. And Superior, 889. So it was okay, but it was always the map length was smaller for Superior, and because we had less markers for Superior that had been mapped. And I think this is because the SNP markers have been biased in certain way, because Atlantic was used for developing these SNP markers. So we expect to have a lot of SNP markers for Atlantic, but maybe not for Superior. I'm comparing here the 2 chromosomes, one chromosome that had the most markers and the other chromosome that had the least. So chromosome 2 had 50 SNPs in Atlantic and 24 in Superior, and chromosome 12, 20 SNPs in Atlantic and 13 only in Superior. So there are differences between how many SNPs we can get for different chromosomes. Another thing I did is a comparison between the physical position and the position in the map. And it has been shown before by Dr. Dauch in his populations. And we see that there is some correlation between the map position and the physical position, but it's not perfect. And we also have this area that it's probably due to this is the centromeric area, so there is less recombination there. And I'm finding the same thing that they found. Well, this is a summary of the map. We have between 71 to 30 markers per chromosome. We have more than twice the number of markers in Atlantic than Superior. The size of the map is also larger in Atlantic than Superior. And the marker positions are not in perfect match with the genetic order in the linkage map. Okay, so now we have a map, a tetraploid map. That's good. But now how do we move to get QTLs? So we need phenotypic data to get that. The traits that I have evaluated are calcium, specific gravity, yield, internal quality, chip quality, and pitted scat. These evaluations have been done in 2 years, basically 2009 and 10. And the data, specific data that I used for mapping was the best linear unbiased predictions, which is number that is like a mean, but not exactly a mean. It gets rid of all the environmental effects and just leaves the genotypic effects. So it's a much better value to use for mapping than just a mean. Well, the procedure in Tetraploid map is associate the trait data. It means load your data file that has the phenotypic information in the software. Then perform the QTL mapping in the software. It's just press a button that says QTL. Then permutation tests, and Tetraploid map does a permutation test with 100 replications. So it's not very, how can I say, we would like to have more than 100, at least 1,000, but it's still okay to find threshold for our LOD values. Well, then after running the permutation test, you're ready to identify your QTLs, whether it's above the threshold. Okay, so this is an example because there are 12 chromosomes and many traits to evaluate, and you get QTLs identified for each parent, and I'm analyzing 2 years separated also together. So it will be many tables if I want to show everything. But I'm just showing chromosome 1 in this table. And you can see that there are, for example, in calcium, we found a QTL in 2010, but not in 2009. But it was present in the overall analysis of both years together. Also, yield, we found one QTL that was present only when we used the overall data set. So you can see there is no QTL that has been present in both years and in the overall data set. It's because we know that we won't find the same QTLs every year because the performance of the clones is not the same. So it's and these traits are affected by genotype, by environment interaction. So, but I wanted to show that in this table. Here I have an example of the calcium QTL in chromosome 1. You can see in 2009 the peak didn't go above the threshold, but we can see a peak. And then in 2010 it just passed the threshold, and in the overall analysis it also passed the threshold. So I consider that as a QTL. And this one comes from Atlantic and is located in the 22 centimorgan position, and the variance explained is 9.9%. And I also found which are the flanking markers for this QTL. So there is one that is at 22.9 centimorgans, and another one at 20.5 centimorgan. And I plotted the phenotypic data using those markers. And we see that there is a difference in the mean values of tubercalcium in this case. However, it's not that clear or that sharp difference, but there is a difference. And also we know that these are quantitative traits, so there are many genes that control these traits. But if we were using something different, like disease resistance, for example, we probably could find more higher difference between the means of the genotypes. Both markers had a negative effect. It means that when the simplex marker was present, the mean went down. And I have another example for yield in chromosome 2. This comes from Superior. In 2009, the peak didn't go above the threshold. In 2010, there was a peak, but it wasn't very clear. But in the overall analysis, we can see that it is a QTL and it goes above the threshold. And this one explained 10.7% of the variance. And I also identified which are the flanking markers for it. And two of them had a negative effect. So when they were present, the mean yield went down. But there was another one that when it was present, the mean went up. So it was a positive effect. And as I have said, there is a lot of information that we can get using this map and using the QTL analysis, and we can discuss a lot about it, but it was just an example that I wanted to show. So using the SolCAP SNPs, I could make a tetraploid map, and I also could use it successfully to identify QTLs. And we can see also that these QTLs are affected by genotype-environment interactions. And well, that's all my presentation. And I want to thank the Hatch grant and the NIFA grant for the support for my studies and also for the research. The University of Wisconsin-Madison and all the researchers there and the graduate students that always help me a lot to get the phenotypic data. I'm really thankful for that. And MSU, David Auches and Joseph Combs that helped me with the linkage phase assignments, that part that is very tedious and time-consuming. Well, thanks to SolCAP for genotyping this population also.
Thank you.